Assalamualaikum and hello everyone. This is SIL 1001 lecture 3 on cell division. So this would be the last lecture with me on the component of cell biology. So after this, starting from next week, you are going to learn new components, molecular genetics with Professor Zulkarnain. Cell division is a process by which a parent cell divides into two or more daughter cells. There are actually three types of cell divisions, three types of cell division. In the eukaryotic system, two distinct types. Number one is, of course, you know this, mitosis. Number two is meiosis. The third one is what we call the fission, cell fission, or the binary fissions. This one in the eukaryotes, and this one is in the prokaryotes. Alright. However, we're not gonna look at this the third type of cell division, and in this video we are going to look at the process of mitosis and also meiosis. The events required for a single round of cell divisions are collectively known as cell cycle. And cell cycle, a single cell cycle, can be divided into interphase and also M. So M stands for mitotic, interphase and M phase. And the interphase, is the phase of the cell cycle in which a typical cell spends most of its life. So during this phase, the cell copies its DNA in preparation for mitosis, and it can be further divided into three G1, S, and G2 phases. In G1 phase, the cell prepare for cell division, in which it will start to synthesize the RNA, protein, for example, the histone and non-histone proteins, enzyme and metabolites, and also duplicate the organelles. And in the S phase, is when the cell, when the DNA replicates and the chromosome duplicate. And during G2 is when the cell prepare for chromosome segregation, in which the cell will synthesize RNA, spindle fiber proteins, and also the condensation of the chromatin will begin at the late of G2. This is to prepare for the process of mitosis. Although we're talking about cell cycle and cell division, just to let you know that there are certain cells that are at what we call G0. This stage, this zero phase, is viewed as either an extended G1 phase, where the cell is neither dividing or preparing to divide. So example of cells that are at this stage is the nerve cells. So these figures illustrate the cell cycle, as I mentioned before, the interphase as well as the mitotic phase. The interphase can be divided into G1, S, and G2. And then next, we are going to look at the process of mitosis at the mitotic phase. So the M phase actually consists of two overlapping processes, the mitosis process itself and also the cytokinesis. You see mitosis as well as the cytokinesis. And mitosis can be further divided into prophase, metaphase, anaphase, and telophase. In the next few slides, we are going to look at the process of mitosis in the animal cells. And we'll start with the interphase. But before that, above you can see the how the cells at different stages of interphase and also mitotic division under the microscope, and down here are the illustration, the cartoon representing the state of the cell. In the interphase, this is the phase when they haven't started to perform cell division. You can see that the chromosomes are significantly less compact. Significantly less compact. Indeed, you can see that they are like intertwined with each other, looking like a spaghetti in a plate. Basically. And then later on in the prophase, this is when you start to see the chromosome, the discrete chromosome structure in which the sister chromatids attach at the centromeres over here. This is sister chromatids. And then you can also see that the nucleolus will start to disappear. And then the duplicated centrosome will separate and then will start to move to the opposite poles. And as well as the formation of the mitotic spindles, which are made of microtubules. And during prometaphase, you can see that the nuclear envelope already broken down, and then the kinetochore form at the opposite side of the centromeres, and as well as you can see that the kinetochores bind to the spindle fibers. For information, kinetochore is actually a disc-shaped protein structure associated with the spindle fiber attached during the cell division to pull the sister chromatid apart. So the kinetochore actually form at the centromere, the center of the sister chromatid over here, what we call the centromere, and link the chromosome to the microtubule polymers. The microtubule polymers that I mentioned is actually the spindles fibers. Next is metaphase. At this stage, the chromosome have maximally condensed, and aligned at the metaphase plate, in the middle of the cell, and the sister chromatids, have started to being pulled away to the opposite poles. As the sister chromatid starts to separated by the loss of the sister chromatid cohesion, the cell then enter the anaphase. So at this stage, this is actually the shortest stage of all of the stages in mitosis. And in this picture over here, you can see that the sister chromatids are being pulled apart. The centromere will be pulled first towards the opposite pole as the kinetochore microtubules shorten. And the final step of mitosis is telophase, during which the nuclear envelope reforms around each set of segregated daughter chromosomes. So at this stage, you can see that the daughter chromosomes already at different opposite poles, and the chromosome have started to uncoil and revert back to the interphase appearance. So at the end, you can see that the cell can be physically separated by separating the shared cytoplasm. of the two daughter cells called the cytokinesis. This is an overview of all of the process, including the interphase as well as the mitotic cell division, including the cytokinesis. In the next few slides over here, 1, 2, 3 over here, just to describe what I have in words, what I have explained before using these images. Now, let's see the importance of mitosis. For the unicellular organism, it is important to increase number of individuals in an organism. This is how they actually propagate. And then you can see that because the mitosis is just, it doesn't involve segregation. Each individual would be genetically identical. And for multicellular organism, for us, for example, as an example, mitosis is important to increase number of cells in individual organism for growth, for replacement of dead cells. Like for example, if you have a cut on your skin, in order to recover, your cells will perform mitosis, to replace the dead cells. And all of the somatic cells are genetically identical. The somatic cells, they don't undergo the second type of cell division, which is the meiosis. They only undergo the mitosis. A second type of eukaryotic cell division is specialized to produce cells that have half the number of chromosomes. So unlike the mitosis I mentioned before, occurs in the somatic cells, yet this one occurs specifically in the reproductive cells. Like for example, the meiosis of the spermatocytes to produce spermatozoa, or the sperm cells, and the meiosis of oocytes to produce ova, to produce the egg cells. So in this type of cell division, in the meiosis, it will produce haploid cells, from the diploid cells. In human, for example, in the diploid state, we have 46 chromosomes, 23 pairs of homologous chromosome. So the N number, the haploid number would be 23. And the 23 chromosomes represent or equals to one complement of 22 autosomes and a sex chromosome. Before I go much further on meiosis, let me first explain to you or introduce to you to the term of karyotypes. A karyotype is the number and appearance of chromosome in the nucleus of a eukaryotic cells. There are actually 4 types of chromosomes. 4 types of chromosome: the telocentric, acrocentric, submetacentric, and metacentrics. So you can see that these different types of chromosome, they are slightly different in terms of appearance. The acrocentric chromosome, for example, is a chromosome in which the centromere is located quite near to the one end of the chromosome. And metacentric is the centromere located in the middle of the chromosome. The telocentric chromosome, this kind of chromosome, the centromere is located at one end, like this. Submetacentric is quite similar to acrocentric chromosome. Is the chromosome in which the centromere is located near the middle of the chromosome. The homologous chromosome, these types of chromosome, this term, shows that chromosome that carry alleles of the same genes in the corresponding loci. For diploid organism, one chromosome, not one pair, one chromosome would come from one parent. Each parent one female and one male. There are also genes that are defined by linked or unlinked genes. Yeah, you're going to learn more about that. I think in the next few slides. There's also terms that you need to know, such as the chromosome and also chromatids. Yeah, I believe that you have actually learned about these points or these terms in your matriculation or during your A-level. This figure shows the human karyotype, yeah, of male and female over here. This is the sex chromosome of female, and here are the sex chromosomes of the male. Yeah, you can see the Y chromosome is smaller compared to the X chromosome, and here are all of the autosomal chromosomes. And they're actually, if they're not arranged like this, yeah, actually under the microscope you can see something like this. Yeah, you can recognize that this is actually a chromosome sets of human female. The cells are the cell is actually frozen at the metaphase. Frozen here, not using the eyes, but actually using this drug called colchicine. You can Google about how this drug work that can stop the cell division at metaphase state. So you can actually see the chromosome at the most condensed state, yeah, where you can easily identify which one is chromosome number 1, which one chromosome number 2, 3, 4, 5, yeah, so on and so forth. In order to view the chromosome, there are several staining methods that can be used. Yeah, for example, the G banding, using GM-star staining. So this kind of banding and G banding produce visible karyotypes that stains the condensed chromosome. So the dark line are actually the dark line over here. You can see there are lines, yeah, with darker color. These dark lines are the AT-rich region, yeah, associated with less active genes. And then the lighter bands are the GC-rich. The karyotyping is important technique, yeah, to find abnormalities in the chromosome numbers. One example is the disease of Down syndrome, in which this individual has extra chromosome of 21. Chromosome 21, so trisomy 21. So instead of 2 chromosomes, this individual of Down syndrome has extra 1 chromosome. So in short, the karyotyping that we've seen before able to reveal certain genetic abnormalities. Other than staining that we've seen before, like this, we can also paint the chromosome, of course not using watercolors, but using in situ hybridization, where fragments are differently labeled with fluorescent markers. So you can see that they can be easily identified based on the coloration of the chromosome. Yeah, pretty amazing and pretty nice, isn't it, to see this kind of chromosome under the microscope. Yeah, this is also another example of karyotyping. Now let's go back to the second type of cell division, which is the meiosis. Meiosis is also known as reduction division. So unlike mitosis, the division process occurs twice. Yeah, 1, 2, and twice. So at the end, of the process, it will produce 4 cells, containing half of the original amount of genetic information. This is just an illustration. You see here we'll start with 4 chromosomes. So at the end, the daughter cells would have 2 chromosomes each. I mentioned just now that the division process occurred twice. The first meiotic division is called the reduction division, and then the second division is called the equational division. Like the mitotic cell cycle, the meiotic cell cycles or meiotic division includes a G1, S, and also G2 phase. And as I mentioned before, the division occur twice. So we have meiosis 1 and also meiosis 2. So similarly like before, for meiosis, we have prophase, metaphase, but just add, one, one, one. So that will indicate the prophase, metaphase, anaphase, telophase state at meiosis 1, and similarly for meiosis 2. Now let's look at the stages of meiosis. So we will start with prophase. I'm not going to explain about interphase. It will be very similar to what I have explained before for the process of mitosis. Prophase 1 is actually a lengthy and also complex process, which can actually be divided into substages, into 5 substages as listed over here. Lactotene, zygotene, pachytene, diplotene, and dikinesis. So these are the 5 stages inside or substages of prophase 1. So the 5 stages, don't get confused with this image because this is a simplified illustration of prophase 1, which are divided into the early, mid, and also late prophase 1. But anyway, coming back to these substages, at lactotene, at this stage, the chromosomes become visible as fine threads, quite similar to the interphase stage. And then at zygotene, zygotene is actually from a Greek word that means paired. So at this stage is where you can see the synapses of homologous chromosome to form what we call the bivalent structure. So this is what I meant by the bivalent forming. You can see the synapses. Synapses begin at the zygotene stage to form the bivalent structure. So these two chromosomes, two homologous chromosomes pairing to form bivalent structure. At pachytene stage is when the crossing over occur to form chiasma or chiasmata. This one, the pachytene. This is also the continuous pairing at several points of the chromosome. So you see the crossing over occurs at pachytene stage. Now let's take a detailed look at the crossing over and also the chiasma or chiasmata formation. So synapses is the formation of the synaptonemal complex, which to align the alleles. So usually the synapses is accompanied by SC or synaptonemal complex. SC it's like a ladder-like structure. A ladder-like structure to align the alleles. So this is the point of where the exchange of genetic material, or what we call the crossing over. So this allows exchange of alleles, which can lead to new combination of linked alleles, and this will lead to genetic diversity. This is how the SC or synaptonemal complex looks like. This is an illustration. You can see that the two regions over here with different colors, red and blue, represent the homologous chromosome, each with 2 sister chromatids. Okay, And the synaptonemal complex is actually important to serve as a scaffold for the crossover, yeah, but it's not required during recombination. Whereas the chiasma or chiasmata is the cytological observation of the crossing over process, yeah, which is clear at diplotin and also dikinesis. Let's coming back to the substages. So we have learned until here the pakitin. So next will be the diplotin, this stage, yeah, in which the bivalent will start to separate, yeah, starting at the centromere. Although I mentioned before that the chiasma starts to occur at the pakitin stage, yeah, the chiasma occur over here, but they become visible at the stage of diplotin and much more at the stage of dikinesis. So at dikinesis, you can see that the bivalents, here is bivalents, are maximally condensed. So at the end of prophase 1, yeah, let's go back here. So at the end of prophase 1, so the nucleoli would disappear and the spindle would form, and the nuclear envelope will break down. Let's review back the prophase 1 stages or substages: leptotin, zygotin, pakitin, diplotin, and dikinesis. Please remember that the chiasma or the crossing over occur here at this pakitin stage, but the chiasmata becomes more visible at the stage of diplotin as well as dikinesis. Yeah, you can see here are the chiasmata, which is the cytological observation of the crossing over or exchange of the alleles or the genetic material. And then moving on to the next stage, which is the metaphase 1, in which the chromosomes are arranged at the metaphase plate. And then at the anaphase 1, you can see that the sister chromatids are being pulled towards the end, towards the opposite pole. And these sister chromatids are from the homologous pair. So the homologous chromosome pairs, this is one homologous chromosome pair, this is one homologous chromosome pair. The pair will be separated, yeah, towards the opposite poles. And at telophase 1, the chromosome, each with two sister chromatid, yeah, would complete the migration to the pole, and you can see that the nuclear membranes have started to form at this stage. So this is the first division, what we call the reductional division, because you notice that it reduced the chromosome number by half. All right. An important point, yeah, I would like to point out that the homologous chromosome pair recombine at this the prophase 1, and then they will be separated, yeah, will be separated or segregate to different nuclei. Next, we have meiosis 2. This is equational division, yeah, where the centromere splits and the sister chromatid separates into different nuclei. Previously, we see that the homologous chromosome pair recombine and segregate, but this time at the second mitotic division or equational division, the centromere splits and the sister chromatid separate. Yeah, so you see the sister chromatid separates. So before this, the homologous chromosome separated. So what you see at the end are haploid daughter cells. So 4 haploid daughter cells that contains half of the genetic content. So the outcome or products of meiosis, I mentioned before, daughter cells, haploid daughter cells. And then secondly, you can see the genetic composition of the daughter cells differ from the mother cells due to two processes. Number one is the random assortment, and number two due to the crossing over that we talked just now during Prophase 1. I'm going to explain about random assortment. Okay, what does it mean by random assortment? How can this lead to different genetic composition between the daughter and also mother cells? During metaphase 1, the non-homologous chromosome are randomly aligned at the metaphase plate. Yeah. Which means this random alignment of the non-homologous chromosome would lead to random assortment at anaphase 1, and which will lead to new combination of chromosomes and therefore non-linked alleles. Like for examples, we're talking about two traits over here, A and B, and then they are two alleles, big A, small a, big B, small b. This is what we call the heterozygous individual. So this heterozygous individual can produce 4 different types of gametes, depending on how the non-homologous chromosomes are aligned. Like for example, big A and big B. We can also see a combination of big A and small b, and not just that, small a and big B, as well as small a and small b gametes. Let me use the next 2 slides to describe about this random assortment. You're talking about A and B still, all right, A and B, and there are actually 2 figures I'm going to show you. A and B. So these are the 2 situations on how the chromosome, non-homologous chromosome, can be aligned at metaphase plate. So situation A, the first situation, you can see that A and B are big A and big B at this site. So during anaphase 1, it will pull these chromosomes, big A and big B. So you would end up to have big A, big B gametes, and also small a and small b gametes. So this is the first situation. Now, let's take a look at the second situation, or situation B. So in this situation, compared to the previous one where big A and big B chromosomes are aligned at one pole, at one side. So this one, you can see that big A chromosome or big A allele at this chromosome aligned with these small b alleles. So you would see that at the end after and during anaphase, and also at the end of the cell division, you can see the production of this combination of gametes, or BA small b, because of this alignment over here. If it's aligned oppositely, you will see this kind of situation. So here in situation B, you see A, big A and small b, and small a and big B. So you would see this kind of combination. It depends on the random assortment of how these non-homologous chromosomes are aligned at the metaphase plate. Now, can you predict how many kinds of gametes can be produced? Previously we only talked about like 2 different alleles, A and B. Let's say there are 3. So how do you know? You can calculate. This is the formula. So if we're talking about 3 genes, you can have 8 different gametes, that can be produced. This is another figure to describe about random assortment, depending on how the non-homologous chromosomes are aligned on the metaphase plate. So you can end up with, you can see different possible haploid gametes combination that can be produced. I've showed you before that if we have 3 genes or 3 pairs of genes, we're going to have 8 different kinds of gametes, possible combination, but we're talking about 23 pairs. So there are actually 8 millions different combination that can be produced. So that's why you see there's no two difference, except for monozygotic twins are genetically the same. Even between siblings, we are very different from each other. Because of this, just one reason, random assortment. And this is just we haven't talked about, crossing over, changing of genetic material between the homologous chromosome that would even contribute more to the diversity. Right now, we're still talking about the different combination, how random assortment can lead to diversity in terms of genetic combination. And I also mentioned to you about the term unlinked genes and also linked genes. So this figure describes about the unlinked genes, which will assort independently. It's just like what I have showed you before in this figure, as well as is in this figure. So you can see different possible haploid gametes formation. For linked genes usually would end up together in the absence of crossing over. If there's no crossing over, if a big A link with big B, you will end up see the same formation or combination in the gametes. This is in the absence of crossing over. So I mentioned briefly about crossing over can also lead to genetic diversity, right? This is the illustration. So remember about the crossing over during Prophase 1. So for this, although I mentioned that the linked genes would end up together, but if there's crossing over, you can actually see different combinations, like for example A and B linked before, but however if there's crossing over, so you see that this part of chromosome would exchange its genetic material. So this particular part over here, exchange genetic material. So you can see that this chromosome would have a recombination, what we call recombination. So you would see big B and also small a would end up in a haploid gametes. Similarly over here, you would see small b and big A. So you can see that this chromosome actually swaps its chromosome segments. And in this illustration, after the meiotic cell division, you would see 4 possible gametes, and the ones in which you see the combination similar to the parental type, the parental type over here would be AB, or AB here. So you call them the parental genotype, this one and also this one. However, if you see something different from the mother cell, from the parental type, you call them the recombinant genotype, like this, right? This is another figure to describe the process of crossing over. You see that over here is chiasma, and these two are chiasmata. This is the site where the homologous chromosomes exchange their genetic material. Without crossing over, only parental types of linked genes can be observed in gametes. With crossing over, parental as well as recombinant types gametes can be produced. Typically, the parental types would would always more than the recombinant types. Now, let's talk about the importance of meiosis. Um, Number one, there are two importance that I would like to highlight in this video, in this lecture. Number one, meiosis ensure gametes are haploid and the zygote will become diploid after fertilization occur. Okay, so please remember that meiosis only occur in the reproductive tissues to produce the egg and also the sperm. And then secondly, the importance of meiosis is to bring about genetic variability through random assortment and crossing over. Very important, because to obtain variation, number one, through random assortment, and number two, through crossing over. And variability here is very important for adaptation to changes. Just imagine that if all of us have the same genetic composition, or in other words, we are clones, meaning that how we respond to environment, to medicine, to whatever stimuli would be exactly the same, right? And just imagine if there's a disease come. Like for example, right now we're talking about COVID, COVID situation. And you know that there are some people would have a different reaction towards the COVID-19 infection, right? Some people would be, the symptoms will be much severe, compared to some other people. If let's say our genotype will be the same, and let's say we can't really stand, or we are susceptible towards the disease, then all of the human race will be wiped out by a single disease. So same goes to plantation, in crops, for plants that are clonally propagated, which means that they are clones, so those plantation would have issues if there's any disease attacked. So usually it will wipe out the whole plantation. If there's like 1 hectare, then 1 hectare will be gone just like that, just by a single disease, for example. So that's why variation, genetic variation is very important for adaptation to changes. Okay, so in the next two slides over here, and this one is just a summary to show the differences between meiosis, mitosis, and also meiosis 1 and also meiosis 2. For meiosis, the cell division occur twice, meiosis 1 and also meiosis 2. You can see here at the metaphase 1, the homologous chromosomes are aligned, and the homologous chromosome pair will be separated during anaphase. On the other hand, in meiosis 2, you can see that instead of homologous pair separated, sister chromatids are separated. And this is what we call the reduction division, because you can see that it reduced already in meiosis 1, the chromosome number was the chromosome number is reduced to half, by 1/2 of the original number. And the second meiosis is called the equational division, right? And in mitosis, if you see that, obviously only one division occur compared to meiosis. And the product itself, the outcome also different. For mitosis, you would end up to have diploid daughter cells, but for meiosis, you would have haploid daughter cells, 4 haploid daughter cells. And I haven't explained this slide, This is about gamete formation. So the gamete formation in male and female is a bit different. For the in male, the spermatocyte, the diploid, will undergo meiosis to produce sperm cells, haploid sperm cells, 4 haploid sperm cells. However, in female, the oocyte, the diploid oocyte, will develop, and undergo meiosis to produce 3 polar bodies, as well as 1 egg cell. Okay, so the product would be, I would like to repeat, 4 haploid cells, sperm cells for the in male, and then 3 polar bodies and 1 egg cells in the female. Okay. All right, so that's the end of cell division. Please contact me if you have any question, and I wish you all the best for your tests as well as for your alternative assessment. Thank you so much for your attention. Take care, everyone.